Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss multiplication of bacteriophage or life cycle of bacteriophage. The bacteriophage can multiply through two different life cycles. They are lytic cycle or virulent cycle. Lytic cycle or virulent cycle. The second type is lysogenic cycle or avirulent type. Avirulent cycle. In this video, we are going to first discuss the lytic cycle or virulent cycle. During lytic cycle of the bacteriophage, the bacterial cell get disintegrated. During the lytic cycle of bacteriophage, the bacterial cell get disintegrated and the progenies of virions are released from the bacterial cell. The progenies of virions are released from the bacterial cell. Then the lytic cycle, which involves 5 major steps. The lytic cycle, which involves 5 major steps, they are adsorption, penetration, synthesis, assembly and maturation, and release. Adsorption and penetration combinedly called as infection phase. Adsorption, penetrations are combinedly called as infection phase. Then synthesis, otherwise called as that synthesis phase is otherwise called as latent or eclipse phase. Then the fourth step, assembly and maturation, that phase is otherwise called as vegetative or mature phase. Then the fifth phase is release or liberation phase. Release or liberation phase. Before we get into the topic, I would like to give you some information. So, what is called bacteriophage? What is called bacteriophage? That virus which can affect the bacteria is called as bacteriophage. So, bacteriophage is nothing but a virus. That virus can affect the bacteria. Here, the virus going to affect a bacteria called E. coli. Bacteria called E. coli. So, a virus which can affect the bacteria called E. coli is called as coliphages. Is called as coliphages. That means the T4 bacteriophage can affect or infect the bacteria E. coli. That is why they are called as coliphages. That is why they are called as coliphages. So, let me explain the process one by one. First, I am going to tell about the adsorption process. So, this is the host cell, that is the bacterial cell. This is the host cell, that is called E. coli cell or bacterial cell, and this is the T4 bacteriophage, and this is the T4 bacteriophage. So, adsorption means the T4 bacteriophage make contact with the bacterial cell wall. Going to make a contact with the bacterial cell wall, and that process is called as adsorption. So, in this process, the T4 bacteriophage tail fibers are very important for that adsorption process, because the tail fibers of the bacteriophage has some chemicals called lipopolysaccharide, and they are act as receptors. They can act as receptor. That means it act as a receptor site, and it recognize the receptor site on the bacterial cell wall. So, the tail fiber act as the receptor site, and they are act to find out the receptor site on the bacterial cell wall. So the tail fibers recognize a receptor site in the bacterial cell wall and it make contact with the bacterial cell wall and that process is called as landing. So the tail fibers of the phage recognize the receptor site at the bacterial cell wall and it make contact between the phage and bacterial cell wall and that process is called landing. Then the contact is made between the phage and bacterial cell wall. After that, the tail fiber bends. The tail fiber bends to anchor and pins, and that basal plate attached to the cell surface of the bacterial cell, the host cell. So after that recognition and the contact was made between the bacteriophage and the bacterial cell wall. After that process, the tail fibers bends to anchor and pins on the bacterial cell wall, and that process is called as pinning. So in the first step, that is adsorption, the tail fiber recognize the receptor site on the bacterial cell wall and it make contact with the bacterial cell wall on the recognition site with the help of lipopolysaccharide, that is the chemical which is present in the tail fiber of bacteriophage, and that process is called landing. And the tail fibers bends and anchor on the bacterial cell wall and that process is called pinning. Then the next step is called penetration. In this process, two major steps are involved. The first one is enzymatic step and another one is mechanical step. So here the bacteriophage is attached to the bacterial cell wall. So the bacteriophage has to inject its phage DNA into the bacterial cell, isn't it? So it has to digest the cell wall of bacterial cell. It has to digest the bacterial cell wall to inject the phage DNA. So this bacteriophage secretes an enzyme called lysozyme. So this is the enzyme which can digest the cell wall of bacteria, on the recognition site. So the bacteriophage secretes an enzyme, lysosome, to digest the cell wall of bacteria. Through the digested cell wall, the phage DNA going to inject into that bacterial cell, So after the digestion of the bacterial cell wall, it has a contracting tail fibers or contracting sheath. So after the digestion, the bacteriophage, the tail sheath get contract, and they converted into strong and thicker, shorter and thicker. So this bacteriophage contract their self and it looks shorter and thicker. At the time, the basal plate get enlarges. For that contraction process, the bacteriophage uses ATP molecule for that contraction. After that contraction, the basal plate get enlarges. Through that enlarged portion, the phage DNA enters into the bacterial cell. Through that enlarged portion, the phage DNA get injected into the bacterial cell without any use of metabolic energy, without using any metabolic energy. So the injection of phage DNA into the bacterial cell, and that process is called as transfection, and that process is called as transfection. The injection of phage DNA into the bacterial cell is called as transfection process, and it called as transfection process. Then the empty protein coat remains outside the host cell. The empty protein coat, so it is the capsid, isn't it? So that protein coat is empty because the phage DNA enters into the bacteria, and they remains outside the host cell, and they look like ghost, and they look like ghost, and they look like ghost. Then the third step, synthesis or latent or eclipse phase. So in this synthesis phase, degradation of bacterial chromosome and protein synthesis and DNA replication takes place. So in the synthesis or latent or eclipse phase, the degradation of bacterial chromosome, then DNA replication and protein synthesis takes place, because the phage DNA takes over the host cell biosynthetic machineries, because the phage DNA takes over the host cell biosynthetic machineries. Then the bacterial genome get inactivated and they break down. The bacterial genome get inactivated and they break down. Then phage DNA suppresses the protein synthesis of bacteria, and it directs to produce the phage protein, So the phage DNA directs the host cell biosynthetic machineries to produce phage protein, and simultaneously the phage DNA get replicated. And that process is called as synthesis or latent or eclipse phase. So in synthesis phase, the bacterial genome get inactivated and they break down. So the phage DNA takes over the biosynthetic machineries of the host cell, and to order to produce the phage protein, simultaneously the phage DNA get replicated, and that process is called synthesis or latent or eclipse phase. Then the fourth step is called assembly and maturation or vegetative or mature phase. So in this phase, the protein coat and the phage DNA are produced separately, isn't it? The protein coat and the phage DNA are synthesized separately, and they are assembled together, and they are assembled to produce phage particles, and the phage DNA and protein coat are assembled to produce phage particles, and that process is called as, and that assembling process is called as maturation, After 20 minutes of infection, the bacteriophage can produce 300 virions. After 20 minutes of infection, the bacteriophage can produce 300 virions. 300 virions are phage particles or bacteriophage particles. So in 20 minutes after the infection, the phage particle can multiply and produced into 300 virions. Then that phage particles are accumulated in the bacterial cell, The phage particles are accumulated in the bacterial cell and they are released by the process lysis, and they are released from the host cell by the process lysis. Lysis means breakdown or burst out. So the accumulated phage particles are released from the bacterial cell by the process lysis. That is why that life cycle is called as lytic cycle or virulent cycle. Virulent cycle means in this cycle the bacteriophages are more virulent. They can actively infect the bacteria. That is why this life cycle is called as lytic cycle or virulent cycle. So that's all about today's video. If you have any doubt, please let me know. Thank you very much for watching this video. Thank you.